Alrighty, good morning. Hopefully this will be a little larger and more clear than the last one. So when I went back and looked, it was a little blurry. So hopefully the screen sharing is working better this time. So we're going to pick up where we left off. Yesterday we talked about the cell theory and the fluid mosaic model and went into the three double membrane bound organelles in the plant cell, which were the nuclear membrane, or the nucleus, the mitochondrion, and the chloroplast. So again, I know some of this is review, but there's a few bits and pieces that that'll be new specifically for plant cells. So okay, again, don't forget we're still talking about eukaryotes since we're with plant cells, but this next piece is going to be the Golgi apparatus. So with your Golgi apparatus, if there's more than one Golgi apparatus, it's generally coined Golgi bodies. So we're no longer going to be referring to double membrane bound organelles. So from here out, they're all going to be single membrane bound organelles. For the Golgi bodies or the Golgi apparatus, actually that's another typo I think there. Sorry. Yeah, that should be sends out, not sounds out. So its job is to send out glycoproteins and polysaccharides. Glyco, sugar, proteins, so the proteins with sugar chains attached to them, and polysaccharides, the complex sugars. So I think I've referred to this as the post office of the cell. So its job is to package up things, designate where they're supposed to go, and then sends them there. So on my picture that I've drawn on the backside of your diagrams or your doodles, I've drawn the Golgi body, and you can see that it kind of looks like a stack of pancakes there, with the smaller one being in the top. So it's just a series of membranes, and so your glycoprotein or your polysaccharide is going to come into the top part, or the smaller area, and is then going to be passed through the membranes, and is going to end up at the very bottom, packaged, and then the Golgi will pinch off a portion of its membrane into what's called a Golgi vesicle, and your glycoprotein or your polysaccharide will be inside of this Golgi vesicle and will be sent wherever it needs to go, whether it's out into the cell somewhere or to be exported through exocytosis outside of the cell. So remember, exocytosis is the active transport of moving something outside the cell. So, your endoplasmic reticulum, yeah, we do abbreviate that with ER. Different types, the smooth endoplasmic reticulum and the rough endoplasmic reticulum. Your smooth endoplasmic reticulum's main job is to make lipids. Your rough endoplasmic reticulum is to make proteins. It's rough because it's got the ribosomes on it. Smooth doesn't have ribosomes. The rough endoplasmic reticulum is going to use vesicles to send the proteins that it's going to synthesize and sending it to the Golgi apparatus to be packaged further and tagged further. So your next thing that you've got is ribosomes. These are the smaller, very, very small structures in the cytoplasm. They can either be in the cytoplasm free floating, or they can be attached to your rough endoplasmic reticulum. And it's important to note that even though they're both ribosomes and they look the exact same, they technically are going to have different jobs. They're going to be responsible for producing different types of proteins. So your ribosomes are primarily made out of ribosomal RNA. Again, a review, but they're made up of that small and large subunit that were separately made in the nucleolus and then passed through those nuclear pores out into the cytoplasm and then reassembled, or assembled rather, because they weren't reassembled in there, but assembled into one large structure, which would be your ribosome. And then your proteins would then be made through protein synthesis. Don't forget, you can also have the ribosomes inside of your plastids, like your chloroplast, your chromoplast, and they can also be found in your mitochondria. So, okay. Vacuoles are storage spaces in cells. Think of them like closets, I guess. Technically, your animal cells do have vacuoles. They are called lysosomes, and those are used to break down wastes. So they're going to contain enzymes that will break down wastes and things like that. And then you technically do have these also in a plant cell. But you've also got other types of vacuoles in a plant cell. So vacuoles are going to have a single membrane that's going to be called the tonoplast. I've drawn that for you on the far right, on that back side of the doodles. You've got some cell sap in there, so it might have other things dissolved in the cell sap, like other nutrients, ions, things of that nature. But this particular guy has a special name. It's called the tonoplast. The membrane is called the tonoplast. In the plant cell, your vacuole generally is going to store things or things that need to be excreted for the plant. Another job that your vacuole can do, so there's different vacuoles, just like there were different plastids, there's different types of vacuoles. It's a very generalized term. But another thing that your vacuoles can do is they can be a deposit for anthocyanin, which is a pigment. This particular pigment is going to be, I'm going to call darker pigments, like purples or deep, deep reds. Like I said, really deep in color. And they can also be a site for crystal deposition. So, with the crystal deposition, in plant cells, oxalic acid is necessary, but you don't want to have too much oxalic acid inside of your plant cell. So, because of that, if you were to get an abundance of oxalic acid, it can bond with calcium and create calcium oxalate. And that calcium oxalate becomes crystallized inside of your cell. There are 3 different shapes that your calcium oxalate can take when it's in its crystalline form. And I've drawn those. It's the last doodle there, but you can have a Druze form, which is kind of like a star, or a feed, which is fairly common, and prismatic. So, oftentimes the central vacuole is the major one, and it's going to take up about 90% of the cell in terms of volume. And again, its primary job is going to store things for the plant, like I said, dissolved salts, sugars, proteins, ions, something like that. And then your cell sap makes up most of that vacuole on the inside. So that would be the water with the dissolved salts, sugars, etc. And it's slightly acidic, which gives it a tart or sour taste. You think like lemons. Lemons are acidic, so they're tart or sour. So, oh, I forgot. If you go back, if you look at the pigment deposit for anthocyanin, where you're going to see this, some trees will turn purple in the fall, and that's because they would have anthocyanin. Instead of stopping the chlorophyll production and then turning on the carotenoid pigment production, some plants will turn on anthocyanin production, and that's why you're going to get some purple trees or purple leaves in the fall. So the anthocyanin is also kind of what gives purple onions their purplish color. So. Okay, so your next thing is the cytoskeleton. These are going to be, your cytoskeleton is going to be made up of microtubules and microfilaments. So again, the protoplast is just the cell without the cell wall. So it makes up the cytoskeleton of that protoplast. Its main functions are going to be to help with cell division. They're also going to help with growth and differentiation of the cell. And the cytoskeleton, so we're not going into specifics yet, but the cytoskeleton as a whole is also responsible for helping organelles move from one spot to another within the cell. Your microtubules are hollow. They're made up of tubulin, of a particular protein called tubulin. Their main function is going to help with cell division. And should your cell have flagella or cilia, it's going to make up the flagella and the cilia. It's going to be the backbone for those two. Parts, they're not technically organelles. Those I would consider them parts. And if you recall from the organelles, when we went over this, cilia and flagella help with locomotion or movement. Your cilia, generally, you're going to have a lot of those. Those are those short hair-like projections that go around the cell, and then the flagella are going to be the longer whip-like. Generally, you don't have a bunch of flagella. You might have 2 or 3, but you can have flagella all over the cell body, but it's not typically. Usually, if you're going to have something going all the way around your cell body, it's going to be cilia. So, microfilaments is the second part of your cytoskeleton, and this is going to be made of a protein called actin. And their job is to help you with some muscle movement. So I don't think we've ever got over it, but if we were to go and break down your skeletal muscle down to its smallest, most simplest version, um, so it'd be like the organelles of the muscle fiber or the muscle cell. You would find that it's made up of actin. And so this is going to help you with your muscle movement, not necessarily in a plant cell, because plants don't have muscle, but for humans. And it's going to help with cell division, and it's going to aid with cytoplasmic streaming, which we'll talk about here in just a second. All right, second to last part is the plant cell. We've got microbodies. These are also single membrane bound structures. There are two different types. We're going to have perioxysomes and glyoxysomes. Perioxysomes, they don't have an inner membrane, but they're going to have their own set of DNA and RNA, DNA and ribosomes. Now, these don't typically fall into the endosymbiotic theory, but what these are going to do, what your perioxysomes are going to do, is they're going to use oxygen to break down fatty acids and amino acids. That's all they're going to do. Your glyoxysomes, usually you're going to find those mostly in your seeds. Not necessarily going to find it in a plant cell. You'd find it in the seeds, the cells that make up your seeds. Mostly because they contain fats and oils that are necessary to convert the fats and oils for germination. So your plant seed can sprout, germinate. All right, and your last one is the cytoplasm. Your cytoplasm is always in motion. Remember, the cytoplasm is the space. The cytosol is what's the fluid, the semi-fluid part that's found inside of your cytoplasm. And so it's always in motion, which that motion is called cytoplasmic streaming or cyclosis. And it's those microfilaments that are going to help with that cytoplasmic streaming, that are going to allow your organelles to be able to move from one place to the other. So this is kind of an overview, a kind of a refresher about the organelles found in plant cells, or just cells in general, but specifically a little bit more into the plant cells themselves. So hopefully it makes sense. If it doesn't, please don't hesitate to ask. And that'll be all on this part. So